Today's presentation is the Fantastic Four: Simcyp rat, mouse, dog, and monkey simulators. Our speaker for today is Dr. Devendra Pade. Devin is currently a senior research scientist in Certara, UK, in the Simcyp division's Modeling and Simulation group. He earned his doctorate in predicting oral drug absorption and preclinical pharmacokinetics from the University of Texas at Austin. His interests include oral drug absorption and the development of animal PBPK models for preclinical species. Devin, welcome to today's event. I'll now turn it over to him to begin the presentation.
So today's webinar, we'll be looking at the Simcyp Animal Simulators, and in the forthcoming slides, I will be giving you first I will be giving you a brief introduction to the slides that I'm going to discuss. So this is a brief summary of the slides to come. Initially, we'll be looking at the introduction to the Simcyp Animal Simulators. And later on, we'll move on to looking at the different models that are included in the animal simulators. And then wherever there is an application case study, we will discuss an application case study in brief. Then we'll look into different kinds of the brain, multi-compartment brain models that we have. We'll also take a look at the different pharmacodynamic models and some toxicity-related models such as the fetal placenta model that we have in the rodent simulators. We'll also talk briefly about the tumor growth inhibition model, which has been recently implemented in the mouse. And then we will also look at some translational tools where you can translate drug PK parameters between species as well as from preclinical species to humans. So in the first section of the presentation, I will first take you to the brief introduction to the animal simulators. So in the Simcyp simulator package itself, there are basically 4 preclinical PBPK models for the preclinical species, and those are basically the rat, which is based on a 250-gram Sprague doggy rat. Then we have the dog model, which is based on a 10-kilogram beagle dog. Then we have the mouse, which is a non-strain specific 25-gram mouse. And we have the monkey model, which is a adult cynomolgus 4-kilogram monkey model. And just to give you a brief background in terms of the potential applications of these PBPK simulators, these are primarily with an aim to kind of reduce, refine, or even in the future, try to replace animal studies. But as of now, as you see, the way drug development takes place, we are moving towards lesser and lesser animal in vivo studies, but still these are not completely avoidable as such. And so as you can see, animal simulators are mainly used to verify these preclinical models, and then basically confirm the assumptions prior to going into human predictions. One of the major applications of these preclinical PBPK simulators is dose estimation for a required plasma concentration time profile or a tissue concentration time profile. Another major application is also to look into the different formulations and different routes of dose administration. And as well as it is also majorly used in toxicity applications, etc., etc. So in terms of the introduction, when I talk about the animal simulators, we are basically looking at the different features here in the different animal simulators, and as you can see here, basically that all the animal simulators have their own species-specific libraries. and each of the animal simulators, they are also accompanied by total body weight flexibility within a certain range. In these animal simulators, you also have models to predict the effective intestinal permeability for orally administered dosage forms, whether it is either from in vitro monolayer assays, or we also have the mechanistic permeability model, which can basically predict the passive permeability in the gut based on QSAR parameters only. And then all of these animal simulators also have the ADME model, or what we call as the Advanced Dissolution, Absorption, and Metabolism model, which is primarily based on the CAT model, but we'll talk about that in the next slides. But it basically enables you to simulate different types of formulations and also food effect studies. Each of these simulators also have inbuilt the permeability limited tissues, wherein you can look at the effect of efflux and uptake transporters in the gut, in the liver, or in the brain, as well as in terms of the elimination options. You have a wide range of elimination or clearance options, such as looking at the disappearance of the drug via the whole organ metabolic clearance, or if you're aware of a certain enzyme that is responsible for the metabolism of your drug, then you can also look at it via the enzyme kinetics option. And again, you can also not only look at the clearance of the drug from the whole body as such, but you can also have clearance assigned in specific tissues, such as not only the liver and the gut, but also the brain, etc. One of the major differences within the simulators is that in the rodent simulators, we do not have any inter-individual variability. So when you are using the rat or the mouse simulator, you are basically simulating a single individual all the time. But in the dog and the monkey simulator, you have inter-individual variability, and hence you can simulate a population. In the monkey model, we also have the PBPK models for therapeutic proteins and monoclonal antibodies, which is basically the biologic section or the large molecules. But I should make you aware that we won't be discussing too much about the biologics part in this seminar. But if anybody is interested in the modeling of therapeutic proteins and monoclonal antibodies, please write to me, and then we can discuss about this more. Each of the simulators come with their own validated compound files and also handy tools such as the batch processor. A batch processor is basically where you can run hundreds of simulations all together at once and create your customized outputs, whereas the other tools such as sensitivity analysis and parameter estimation are also available in all the animal simulators. So when we are talking about these different types of PBPK models for each of the species, this is an example of a recently published paper by our colleagues on the rat PBPK model. And it basically highlights the kind of constraints and the kind of limitations that one faces when you're trying to develop an in-depth PBPK model for a certain species, because we assume that because it might be a rat species, so there might be tons and tons of data. But if you go through this paper, it will essentially highlight what difficulties would one face in order to get an as accurate as possible physiologically-based pharmacokinetic model for a certain species. And I would encourage you to go through this paper. And I highlight here because based on this last point here, due to the in-depth meta-analysis that undergoes in building each of our animal models or even the human model, more and more pharmaceutical companies are now coming back to us with regards to wanting to know the kind of meta-analysis that we do, and they are basically harmonizing their internal preclinical databases based on the meta-analysis of each and every physiological and anatomical value that we are using in our simulators. So I think this is an important point to be mentioned, because we do take pride in our meta-analysis and the effort that goes into our meta-analysis, and we are happy that we are able to build such robust PBPK models. So when looking at the overall architecture of the animal simulators or even the Simcyp simulator, the architecture kind of is similar, where you can see that we divide the simulator into three parts, wherein you have the systems data, which is basically the species data and the species library, the physiological and anatomical parameters, the enzyme abundances, etc. And then you have the trial design, and then you have the drug data. So basically, the systems data and the drug data are combined in order to get a combined in the mechanistic IVIVE and PBPK models, and the trial design together enables you to predict pharmacokinetic concentration time profiles in plasma and tissues, as well as drive your connected PD models. But when it comes to species, when you're talking about these species parameters and the systems approach, etc., when you're looking at the animal simulators or animal PBPK models, one of the major applications is basically translating your PK in between species and from animal species to humans. So when you're translating PK profiles, or when you're translating data from animals to humans, you have to keep in mind that there are certain parameters and certain quantities that can be basically translated readily from animals to humans. And because of this systems approach that we have, if your model is pretty robust in terms of its input parameters, then of course you can switch between the species or within a species. Say, for example, you're looking at a mouse PBPK model, and you have a wild-type mouse, then with a verified drug model, then you can switch from a wild-type mouse to a knockout, say a PGP knockout mouse model, and then you can look at the predicted PK profile of your certain drug with the in the knockout PGP model as well. And so on the right here, you can see in certain parameters that are listed for cross-species translation. So for example, your drug physicochemical parameters, or let's say your intrinsic permeability in the intestine, or the partitioning of the drug into the tissues, etc. These are certain parameters that can be readily translated. Maybe sometimes the whole organ metabolic clearance can be translated, but I'm not talking about enzyme kinetics here. So so you can see here there are some parameters that can be translated. But then when you're looking at other parameters, then you have to step back and think about it, because every species has their own differences. Say, for example, the metabolizing enzymes are different in each of the animal species, because CYP3A4 in the human may not be available as an enzyme in the dog or the monkey, or the rat or mouse. But then there might be equivalent enzymes for CYP3A4, and you have to know which are those equivalent enzymes and what is the activity of those enzymes. Similarly, it's the case for the transporters as well. Apart from that, when you look at the drug parameters, the situation becomes even more complicated, because when you're looking at fraction unbound in plasma, then because of the differences in the abundances of these proteins in different species plasma, your FU plasma is going to be different. And so similarly, your blood to plasma ratio, or as I was talking about, because the enzymes are different, your metabolism itself for the drug can be different in terms of the Vmax and the Km, as well as for the transporters, the Jmax and the Km can be very, very different. So when you're talking about translating from animals to humans, you have to basically think about it in terms of what parameters can be extrapolated to human and what parameters cannot be extrapolated to humans. So this is one of the reasons why automated translation or direct translation without is not possible, because on the plot here you can see on the y-axis you have the human absolute bioavailability, and on the x-axis you can see that we have the animal bioavailability, which comes from different species such as the mouse, rat, dog, and the primates. And you can see that absolutely there is no correlation at all, and none of the species can predict the human bioavailability correctly. And one of the reasons for that is the underlying different types of metabolism and the different plasma protein binding and the different blood to plasma ratio, which is responsible for this lack of correlation. And as I said, there are some parameters that can be translated. So for example, when you look at the effective permeability here, this is from Zakir Milani, and you can see that on the y-axis you have the effective permeability in the rat determined by single pass intestinal perfusion, and similarly you have on the y-axis the human effective permeability determined by the Loki gut. And you can see that there is quite a good correlation between the rat PEF and the human PEF, though you can see that there is some sigmoidal relationship here which is going on, but at the same time nevertheless they are very, very well correlated. So properties such as intrinsic permeability can be translated from one species to another. And here is another plot from the same paper where they have tried to look at the prediction of the fraction of dose absorbed in the human on the y-axis here from the effective permeability in the rat. So as you can see here, there are certain positives about translating from one species to another, whereas if you look at that absolute bioavailability graph, then it's very difficult to figure out how the whole thing can be translated. So now we move on to explaining in brief what the ADAM model, or what we call as the Advanced Dissolution, Absorption and Metabolism model, and we'll discuss a application case study in brief in the dog model. So as you can see here, in the ADAM model, basically this basically divides the GI tract into 9 different compartments, and from the stomach to the colon. And this schematic here represents the dog ADAM model. So as you can see here, the jejunum is consisting of 5 parts, and there is 1 duodenum and 1 ileum and 1 colon. And the jejunum is divided into 5 parts because the dog jejunum is much longer as compared to the other GI segments. And of course, each of these compartments have their own segregated blood flows, etc. I won't go into the depth of the ADAM model, but most of the specifics can be looked at, can be read from this paper by Jamai et al. In 2009. So in use of the ADAM model, I will be going through a recently published example in the next couple of slides. This was an example of modeling the oral absorption of Danazol in the fasted dog using the Simcyp dog model. And one of the highlights of this work was that this work comes out from the collaboration with the FDA, Center for Veterinary Medicine, and Simcyp basically has a CRADA with the FDA. And what we did in this work was that we looked at modeling 9 different oral formulations of Danazol, which basically consisted of solution formulations, suspension formulations, and immediate release formulations. And the main goal of this work was to basically highlight the interaction that goes between a drug like Danazol and the type of formulation that is being used for administering the Danazol, as well as the interaction between the drug, the formulation, and the GI physiology of the species where the drug is being tested. So as you can see here, this is the first example where we were looking at a commercially available Danazol formulation, which is Danocrine 100 milligrams capsule, and the model was built basically using all QSAR data and in vitro data inputs. So basically, it was a completely bottom-up model, and it basically highlighted all the interaction between the drug formulation and the gastrointestinal physiology. And as you can see here, the blue line here is the predicted data, and the black dots are basically the the observed profiles along with the variability, which is shown in as standard deviation. And considering that it's a completely bottom-up model, it was a very good, robust prediction, and the model was also proved to be robust because I'm just highlighting here 2 formulations, but on the overall, we tested 9 formulations and we had successful predictions for that. This is another formulation that we looked at where danazol was dissolved in a surfactant called labrafil, which is known to undergo lipolysis. Basically, lipolysis is the breakdown of the surfactant to release mixed micelles. And due to the release of the mixed micelles, danazol tended to partition more into these mixed micelles, and there is a counterbalance or counteracting effect wherein because of the micelles, danazol dissolves more, but at the same time, because of the increased partitioning into the micelles, there is a negative effect on the permeability. And hence, as you can see here, basically the red line is where, if you can see the black dotted line is basically when danazol was simulated as a solution where it over-predicted, but then when it was, when lipolysis was taken into consideration and precipitation was taken into consideration, then the model predicted well. So these are some subtle nuances which you can highlight via modeling and simulation, and which you would probably not be able to figure out via if you don't have correctly designed in vitro or in vivo experiments. The next that we will be looking at is the multi-compartment brain model that we have in all the animal species. And the multi-compartment brain model is basically a part of the overall PBPK framework, wherein you can see that all the tissues are connected to each other via the arterial and the venous blood flows, and the permeability limited tissues, or the tissues wherein you can model the effect of transporters, are lined by a dark border. And as you can see here, the brain is basically lined by the green border, which signifies that it's a permeability limited tissue. So in the animal models, we basically have two types of the multi-compartment brain model. So in the rat and the mouse, or in the rodents, we basically have a 3-compartment brain model, which divides the overall brain into the CSF compartment, the brain mass compartment, and the brain blood compartment. Whereas in the dog and the monkey model, we basically have the CSF, which is further divided into the spinal CSF and the cranial CSF, and the brain mass compartment is divided into the intracellular fluid compartment and the interstitial fluid compartment. So basically, you can see here that we have a multi-compartment mechanistic brain model, and as you can see, the red lines or the arrows basically indicate the flow of fluid from one compartment to another, and the blue arrows basically signify either the passive permeability, which is basically the double-headed arrows, and the single-headed arrows signify the uptake or the efflux of the drug from either of the compartments. So in terms of populating such kind of complex models, we basically strive to populate these models with as much physiological and anatomical data that is available in the literature. And again, I come back to our efforts for the in-depth meta-analysis. And as you can see here, all of the physiological and anatomical parameters that are involved in the brain compartment, we have species-specific values for each of the parameters, such as the volume of the intracranial blood, or the flow rate of the cranial and the spinal CSF, the pH of the different compartments, and the amount of protein in the different compartments. And then when we look at the types of transporters, then all the compartmental models are equipped with the apical and apical transporters for the blood-brain barrier and the blood-cerebrospinal fluid barrier. And as you can see here, the yellow arrow basically marks that currently only relative values for the abundances of the transporters are incorporated. because all through the years there wasn't enough absolute abundance data, but now, as you can see, there is more and more absolute abundance data being developed. And this is one recent publication that comes from the result of the grant that Simcyp provides to different universities. And the recent grant was given to the University of Manchester, and they basically are working on the determination of the absolute abundances of the blood-brain barrier transporters in different species. And this is one of the recent results that have come out from them. And these are the absolute abundances of different transporters in the rat brain microvessels, which will soon be incorporated in the Simcyp Animal Simulators. Now, when you're looking at modeling the kinetics of a drug in the brain using the brain model, it is important to know what kind of drug data is needed. So basically, when you're looking at these different compartments, you are the first thing that should come to your mind is basically the binding of the drug to the different brain constituents, such as the brain tissue, or binding of the drug to the cerebrospinal fluid. And the other important factor is the transfer of the drug from one compartment to another, which can be either passively, which is determined by the permeability surface area product, or PS as we call it. So the PS across the BBB, or the blood cerebrospinal fluid barrier, or you need to know the transporter clearance via the Jmax and the Km through the different barriers of the brain. So when you're talking about the binding of the drug to the different brain constituents, this is one reference from Leedy from Pfizer, and basically what you need to know from this is that if you should strive to get binding of your drug in the species of your interest, but it is not possible to every time to get that data. And hence, this slide basically signifies that if you don't have brain tissue drug binding in a particular species, even then you can have data from another species, because all of these species, in terms of the brain tissue binding, they are very tightly correlated. And so you can see here there are different species here, and the paper basically says that all of these, so the brain tissue binding is interchangeable between the species. Apart from that, in the simulators, we also have automated tools in order to predict the passive permeability surface area product. These tools are primarily based on QSAR and animal data, and so caution should be advised when you are using these predictors. But nevertheless, if you don't have in vitro data, then these QSAR prediction tools are pretty handy in doing your modeling. Next is we will briefly talk about the pharmacodynamic models that are present in the animal simulators. And one of the highlights for having pharmacodynamic models in these simulators, in the animal simulators, is that when you have a verified PK model, then what you can do is you can, once you have verified your PK model, you can predict the different tissue concentration time profiles in your particular species. And these predicted PBPK tissue concentration time profiles then can be used to drive your PD models. And one of the advantages of that is that when you're using these tissue concentration time profiles to drive your PD models, you're essentially using the concentration time profile or the amount of drug at the site of action to drive your PD models. So hence you can see here, if you have a verified PBPK model, then you can use a range, you have a choice of a range of tissue concentration time profiles, right from the brain tissue, or even if you want to drive your PD model using luminal concentrations, intestinal luminal concentrations of your drug, then this can be done via the PBPK model. And within a tissue also, you can use the tissue internal concentration, such as the intracellular fluid of the brain or the spinal CSF of the brain concentration to drive your PD model. In terms of the different types of PD models that we have the basic direct response models, which involve the simple Emacs and the sigmoidal Emacs models, as well as we have the classic indirect response models based on the Daneka paper, which are basically the inhibition of production and loss of response and the stimulation of production and loss of response. Apart from that, you can also use the Lua scripting or the PD custom model in order to write your own PD script and use the PBPK predicted concentration time profiles to drive your scripted PD model. So the overall potential application of having this is that from your preclinical species, you have a verified PK model, and then you can use any of these tissue concentration time profiles to drive your PD models. And apart from that, you can also include, say for the rat and for the dog and the monkey models, you can include, because of their inter-individual variability capacity, you can include variability from the CYP and transporter phenotypes in order to drive your PD models, which can essentially make prediction more efficient. Next, from the pharmacodynamic models, we will move on to looking at the rodent fetal-placenta model. And the development of this model comes from basically Simcyp is a part of this Eurotox risk grant, which is basically a huge grant on the European subcontinent here. In terms of looking at the toxicity of different drugs and chemicals in preclinical species. And the fetal-placenta model in the rat and the mouse model is basically here looks at the maternal blood, the placental chamber, the fetus chamber, and the amniotic fluid chamber. And some of the key assumptions are here is that you can account for the passive and the active transport of the unbound drug through these different compartments. And you can also account for the total litter size in the fetus, as well as you can also account for the metabolism of the drug or the chemical from the fetus compartment. And this is one of the applications that was presented by one of my colleagues, Oliver, at the general meeting of the Eurotox risk last year. And this is a model for the teratogenicity of valproic acid, which was developed in the mice fetal-placental model. And this basically looked at the effect of valproic acid on the mice fetus at day 8 gestation time point. And as you can see here, the green line basically shows the prediction of valproic acid in the maternal blood of the mice, and the yellow line basically signifies the concentration time profile of valproic acid in the fetus of the mice, and the dots are representing the observed data, and you can see that the model is predicting pretty well. So in terms of the ratio of the embryo to maternal concentration ratio of valproic acid, this also seems to be predicted pretty okay in terms of maintaining the steady state and simulating the effect of valproic acid and the partitioning of the drug between the embryo and the maternal blood. Next is we are looking at the tumor growth inhibition model, which was implemented in the mouse model. And the tumor growth inhibition model here, which was implemented in the mouse model last year, it basically looks at the incorporation of the tumor compartment within the overall minimal PBPK framework or the overall full PBPK framework. And depending on the location of the tumor, the physiology of the tumor is taken into consideration. Say, for example, if the location of the tumor is along with one of these tissues, then the composition of the tumor resembles those tissues. And if it is with the blood, if it is with the lung, then it basically resembles the lung physiology. So this is one of the potential verification of the tumor model in the mouse, and this was presented by Dr. Abdul Jalil recently at the PAGE meeting in Sweden. And this was a verification of erlotinib and its effect on the growth of the tumor in the mouse at a dose of 25 milligrams per kilogram. As you can see here, on the y-axis is the tumor volume, and the red dots are the observed tumor volumes, and the blue line basically shows the growth of the tumor as modeled by the tumor growth inhibition model. In this particular example, the Simeoni model with 3 compartments was used to look at the growth of the tumor in the presence of erlotinib. And then the same model, it was extrapolated to the 100 milligrams per kilogram dose to see the effect of the drug on the inhibition of tumor growth. And then here you can see from the model itself, you can predict the plasma concentrations of erlotinib, the concentration of erlotinib in the whole tumor tissue, and then the concentration of erlotinib in the tumor intracellular water. So as you can see, it's a fairly mechanistic model wherein you are looking at the concentration of the drug in the different compartments of the tumor, such as the intracellular as well as the extracellular water, etc. Now we move on to the last part of the presentation, which is basically the translational tools, and we'll briefly talk about the translational tools that can be used to translate data between species and to humans. So one of the translational tools is the use of the McPEF model, wherein, as I said, you can translate the intrinsic transcellular permeability from preclinical species or within the preclinical species as well as from animals to humans. And this intrinsic passive transcellular permeability. So generally in our everyday projects, we have access to the monolayer Keiko2 permeability. And so in many of our models, you can see that you can predict from a monolayer apparent permeability to an effective permeability using the apparent permeability to the effective permeability correlation. And so you can basically predict the effective permeability in a particular species from your Keiko2 or your apparent permeability. And then the next step would be to move from this apparent permeability mode or the Keiko2 permeability prediction mode to the mechanistic permeability mode, where you can basically adjust the intrinsic transcellular permeability value to match your effective permeability value. And once you have matched this effective permeability value, this value of the intrinsic transcellular permeability now can be transferred between species in order to predict the permeability in a different species. Now, what this allows you is that there are 2 advantages to it. One advantage is that you can transfer this intrinsic transcellular permeability from one species to another. At the same time, when you're using this intrinsic transcellular permeability, it will predict the regional permeability in the small intestine, because we are all aware that the permeability throughout the small intestine is not the same value, and it will change because in the duodenum, the permeability is known to be much higher as compared to the ileum and to the colon. So this way, you can actually move from one species to another in order to predict the passive permeability for your drug. Also, if you don't have a handle on the intrinsic transcellular permeability, this can be predicted using the inbuilt calculator, where you can predict the intrinsic transcellular permeability from the octanol water partition coefficient, or you can determine it via in vitro designed experiments. Again, I would like to note that Simcyp is currently working on the SIVA module 4, wherein you can basically input your monolayer Keiko2 permeability data in order to extract the intrinsic transcellular permeability. Another translational tool is the use of KP scalar in order to predict the volume of distribution at steady state. And if you look into the human simulator in the prediction toolbox, there is what we call as the KP scalar from species to human. And here what you can do is that you input it picks up the drug physicochemical parameters from the compound that you have built into the simulator, and then you basically choose the species of interest, and then you basically adjust the KP scalar in order to have a value of predicted VSS that you're looking for. And then when you click apply, this KP scalar from that particular species gets applied to the human simulator in order to predict the volume of distribution in the human. And hence this kind of is a very handy tool wherein you can look at the effect of the animal KP scalar on the prediction of VSS in the human. Another important tool in the animal simulators is basically the calculation of the retrograde intrinsic clearance from the in vivo clearance value. And this retrograde model is now implemented in all the 4 animal simulators. And you can basically input your in vivo intravenous clearance or your in vivo oral clearance along with the information on FA and FG in order to predict your enzymatic clearance if you are aware of a particular enzyme that is responsible for the metabolism of your drug. And if not that, then you can also predict the intrinsic clearance for the whole organ, such as the liver or for the intestine. So that was a gist of the different features of the animal simulators. I have not gone through the depths of each of these because it is very difficult to go through each and every detail. But if you have any questions, do write to me. My email is here. And I would like to acknowledge the help of my colleagues, of course, at Certara, as well as acknowledge the help of Dr. Marlene Martinez, with whom we have the CREDA and due to which we have been working on various projects. So now I open the panel for questions, and I'm happy to answer any questions if there are.
Great, thank you so much, Devin. As Devin mentioned, we encourage our audience members at this time to enter your questions for Devin into the Q&A box. It looks like we have our first question. Animal simulators are part of the Simcyp simulator installation. Can these animal simulators be installed separately from the human simulator on a different machine?
Hi. Yeah. Yes. Of course. Right now, you can. So if you have a PC-based license or something like that, you can install the human simulator on one machine and then the animal simulators in a different department on a different machine. Yeah, definitely. These installations can definitely be separated now, because this has been done based on feedback from many consortium members where they were asking that because the animal simulators are used more widely in a different department as compared to the human simulator. So yes, these can be installed separately.
In the Danazol case study example, what are the other formulations that were modeled?
Oh, yes. So in the Danazol case study, I have a slide here. So these were the different types of formulations that were actually modeled here. we basically looked at a bunch of oral solution formulations, oral suspension formulations, and IR capsule formulations. These basically were of different doses, and they were all taken from different in vivo published results. And that's the reason I was saying the model is pretty robust, because it was able to predict all of these formulations. Yeah.
Someone wants to know, is it possible to model biologics like monoclonal antibodies, PK? If so, can you do full PBPK with a TMDD, target-mediated drug disposition? Is that possible?
Yes, definitely. So in the animals, it is the monkey simulator which has the therapeutic proteins and monoclonal antibody models implemented, and you can do a full PBPK modeling of either a therapeutic protein or a monoclonal antibody along with TMDD in the monkey simulator. Yes. Of course, in the human simulator, you already have the biologics module, but within the animals, it is the monkey simulator that you can look at the disposition of biologics.
Do you have face any challenges predicting the permeability of drugs with poor solubility and very high protein binding?
That's a good question, but yes, I don't have a example right in the top of my head. I think with poor permeability and high protein binding, well, I can't recollect of an example right on the top of my head, but there have been reports where it has been published, it has been predicted pretty well as well. So.
One of our attendees remarks that some tumors
it is challenging.
Oh, sorry, go ahead.
No, I was saying it is challenging. That's what I'm trying to say here, that definitely it is challenging. Because also, I think I would refer back to one of my previous papers on the prediction of permeability in the rodents, in the rat and the mouse. I published a paper 2 years back, and within the range of, we looked at more than 50 compounds to predict the permeability, in the rat and the mouse intestine using the MECPEF model. And what we observed in that is that for moderately and highly permeating drugs, the model was very, very good in predicting the passive permeability. But when you look at the extremely low permeable compounds, there is a lot of disagreement between the predicted and the observed data. So yes, I can't recollect exactly the protein binding part of that, but I definitely know that there was a bunch of compounds which were low permeability compounds, and yes, there was a bit of disagreement between the predicted and the observed data.
Some solid tumors have diffusional characteristics that are markedly different from healthy tissue. Is there a way to accommodate this fact, because this has been a significant challenge in drug delivery for certain solid tumors?
Can you repeat the question? Sorry.
Right. What I think she's saying is often certain solid tumors, we can't treat the cancer because there's difficulty in getting the drug to the solid tumor, and that's potentially because these tumors, the diffusion characteristics for drugs within the tumor is different from healthy tissue, and she wanted to know if there was a way to accommodate this difference between healthy tissue and tumor.
Oh yes, definitely, because the tumor models that have been implemented in the mouse or even in the human, they are basically representative of the actual tumor composition. So as I said earlier, our meta-analysis is always in depth, and so if you look at the tissue composition of the tumor itself within the models that have been implemented, you will see that they are they are characteristically different from the healthy tissue, and hence whatever tumor tissue partitioning that happens will rely on this different tumor composition, and hence you will be able to predict the representative concentration in the tumor itself. Yeah.
Devin, this looks to be our last question from the audience. Do you have an example on the use of the multi-compartment brain model? Are there plans to include the absolute abundances of transporters in the brain model?
Yes. For the animal simulators, we currently do not have a published example in a peer-reviewed journal for the effect of or to look at the pharmacokinetics of a drug in the animal brain model. But in terms of the absolute abundances, as I said during my presentation, we are now collecting data on absolute abundances for different animal species, and most likely we will be looking into implementing these absolute abundances sometime in the future.